Hello, my name is Fernanda Rodriguez, and I've been an intern for Dr. Stomova in her lab. I will be talking about sex differences in the peripheral blood gene co-expression networks following intracerebral hemorrhage. First, to define what ICH is. Intracerebral hemorrhage, or ICH stroke, will then elicit an immune response, and our study wants to look at the sexual differences between male and female subjects. We utilize transcriptomic analysis, which can illuminate on the transcriptomic architecture of these patients. In order to do so, we use an Affymetrix HTA 2.0 microarray used to gather whole genome RNA expression on peripheral blood. We conducted this on 24 male and 9 female ICH subjects and 24 male patients with a complementary 24 male and 9 female vascular risk factor controls. We then used the RNA expression levels for weighted gene co-expression network analysis, or WGCNA, which is an R script that helps construct co-expression modules. And then from these co-expression modules, we found modules that were statistically significant to diagnoses, and we then put these significant modules into David gene ontology to find statistically significant biological pathways and molecular functions. Furthermore, we used ingenuity pathway analysis, IPA, to describe the biological pathways and predict downstream effects. Next, to explain how WGCNA works. First, what is a co-expression network? Co-expression networks are groups of genes that show similar patterns of expression. For example, if we had this single patient with gene light blue and gene dark blue, they both have similar low activity and similar high activity in different patients, and therefore they would be grouped together. This analysis is done on a group of genes and not a gene by gene basis. Furthermore, these modules are only correlations and do not show any explicit biological significance, therefore the need for David or IPA to determine biological significance. Once we construct these co-expression networks, we then go into hierarchical clustering. Hierarchical clustering has two major ways of calculating, which is the hard threshold or the soft threshold. If we look towards this diagram, the black line delineates the hard threshold, which is essentially a binary decision. Either it's 0 or 1, delineated by this equation. However, a soft threshold is a continuous adjacency, as described by these colored lines. And since the continuous adjacency function analyses, we chose to use the soft thresholding. And in doing so, we have to set the parameters of the beta. And to do so, we have to balance our R squared value and our mean connectivity. Once we assign our beta value, we then go into constructing the dendrogram. And in order to do so, we utilize a dis or similarity matrix. For a quick example, we have this matrix where genes 1 and 2 are represented as closely related and having this deep red color. And so that translates to them being branched together at a singular node on the dendrogram. Then, if we go into gene 3, it's a lighter red, making 3 being one node above the 1 and 2 node, and so on and so forth, with white delineating very dissimilar objects, where we have gene 1 and gene 9 having a very white color, and therefore very far from each other on the dendrogram. For our WGCNA, we conducted two, one for male and one for females. And these dendrograms can be seen here. The male dendrograms have 40 discrete co-expression modules, with 2 of those being significant, outlined by the stars. The female WGCNA analysis outlined 29 discrete co-expression modules, with 5 of those being significant, noted by these stars. Although these modules might seem small, most of these modules are at least 500 genes, with this turquoise module being over 2,000 genes. So once we have the list of genes in the significant modules, we then inputted these significant modules into David gene ontology. And our gene ontology analysis for males yielded 45 gene ontology terms that passed Benjamin Hopman's multiple testing, with 172 gene ontology terms passed for females. For the gene ontology terms, there are only 8 overlaps between male and female ICH response, which shows that just on gene ontology analysis, there's large differences between male and females. First, looking at male-specific gene ontology terms. Some highlights are the Fc epsilon receptor signaling pathway, the B-cell receptor signaling pathway, and immunoglobulin receptor binding. Most of these pathways were in relation to the immunoglobulin response. First, onto some background on what immunoglobulin responses are. Immunoglobulins are typically antibodies, and these antibodies have a heavy chain and a light chain. And we'll see that in the male gene ontology terms, these regions of the antibody are highly regulated. Now to look onto some of the female ICH gene ontology terms. Some highlights are the T-cell receptor complex, the RAGE receptor binding, the phagocytic vessel membrane, apoptotic process, and inflammatory response. Although these ICH responses are largely different, we do find a few gene ontology terms that are similar. The ones we'll be looking at primarily are blood coagulation and innate immune response. But these highlight, so even when they're showing the same gene ontology term, the individual genes are often different. But the story gets even deeper. When we look at the network of blood coagulation, we see different expression patterns and different molecular targets. First, to look at the similar genes. There's 4 genes that overlap, and the circles represent similar expression patterns. So in the male network, the capsid A1 and capsid A2 are upregulated, similar with the female network. However, the gene overlaps of H3CR and H3C6 are downregulated for the male network, but upregulated in the female network. And if we also look at the network as a whole, the male network is predominantly downregulated, with the female network being predominantly upregulated, showing that it's the same gene ontology term that was significant for diagnoses, but they had different expression directions. Furthermore, in the female network, there's a large array of different molecular targets. In the male network, you predominantly only see proteins, which are represented by these circles, and only 2 transporter genes represented by these circle and the rectangle. However, in the female network, there's a large diversity in the genes in the molecular targets. However, in the female networks, there's a large diversity in the molecular targets. These represent ion channels, these represent transmembrane signaling, these represent enzymes, these represent various proteins, these are kinases. We'll see a similar story by looking at the innate immune response in ICH, with the male network being predominantly downregulated, with all of the female network being upregulated. Next, to look at our pathway analysis. First, onto the male-specific pathway response, the ICH. 2 of the strongly correlated pathways were receptor-mediated endocytosis and classical complement pathways. Here, we see a very similar story of downregulation of male genes, with primary targets being immunoglobulin regions, with these being the light chain regions and these being the heavy chain regions. Next, to look at the female-specific pathways. We have inflammatory response and degranulation. Here, we see a large diversity in the molecular targets from transmembrane receptors, enzymes, proteins, peptidases, kinases, which is a strong contrast from our male network that primarily target immunoglobulin regions. Additionally, we see in these specific pathways associated with female ICH response, we see lots of upregulation of genes, whereas we saw in the males downregulation. Now onto the similar pathway found through IPA, which is the B-cell receptor signaling. We have similar predicted suppression in both pathways. However, these only represent a single module. Furthermore, in a separate female module, we see predicted activation of the whole pathway, which demonstrates that females have networks that can both upregulate and downregulate the signaling pathway. Male ICH response only had predicted downregulation. It implies that there's added levels of transcriptional architecture in the female ICH response. Onto another common signaling pathway, endocytosis. Here we finally start to see some similarities in the diversity of molecular targets, where we have the male network having transmembrane receptors, enzymes, a large array of proteins. However, we still see that these are primarily targeting immunoglobulin genes, whereas the female network still has a large arrays of kinases, enzymes, transmembrane proteins, and has increased activity with the majority of the male network being downregulated. By taking a look at these different pathways, we seem to be coming to the same conclusion, is that there are some molecular function and signaling pathways that overlap between male and female ICH response, and they even have overlap in the genes. However, the individual genes in the pathway are often in opposite directions, with males being downregulated and females being upregulated. Transcriptional regulation, which demonstrates our need to understand the sexual differences in ICH immune response. Thank you. Now, here are some references for diagrams that I presented earlier in the paper. Here are the legends and annotation notes for some of the diagrams that I created through IPA. I'd like to give a very special thank you to Dr. Stamova and Bodhi for helping me through this process. They have been amazing mentors in teaching me how to work through WGCNA, IPA, and striving towards personalized medicine and understanding sexual differences and trying to dive into the large complexity of all these networks that we're trying to look at. And here's our funding disclosures. Thank you.